Well, hello everybody. What we focus on in our lab is really production strategies for the production of therapeutics and complex biologics. Complex biologics being something that's a little bit more complicated than a drug that's produced by biological means. When you talk about regular drugs, it would easily, it'd be easy to go to a generic because there's only one way to define the properties of that drug. When you go to a complex biologic, it becomes a little bit more difficult because there's many many aspects that can determine the quality of the final product. And so these are things that we're studying in our lab. So really, it can be summed up as simply as what can cells do and what do cells need so that we can end up controlling them. We truly believe that cells follow orders, but they can only do what they can do. And what I mean by that is that depending on what's at their disposal, they can do different things. And so if we know what they can do from a genetic standpoint, because they've got the instructions to be able to do it, then once we know that they have that instruction set, can we provide them the required elements so that they can make the pudding, as an example. So the goal is to understand what these different types of cells can do and under what conditions. One way that we're going about doing this is using a metabolomics approach, metabolomics being the global study of all metabolite changes in a biological system. Now, that's a very ambitious statement, and there's no way to do that, and there's no true metabolomic method that can give you the entirety of all metabolites in a biological system. So we focus on one type, which gives us a subset of all those metabolites, but is actually a quite powerful tool that gives us a lot of information, and that's based on NMR. And not only do we use NMR, but we use targeted profiling. So from the spectra that we achieve from examining our different samples, whether they be supernatant samples from cultured cells or extracts from cells, we take that and we deconvolute that spectra, because the nice thing about these spectra is that they're additive, and the sum of all spectra will give you the observed spectra. And from that we can actually pull out really nice profiles, as seen in the bottom right hand corner. This is just a subset of all the metabolites that we can measure. It's 3 to 4 times that quantity that we usually get out of cultured cells, and you can see that we even are able to get a 95% confidence interval on our measurements, and we have a pretty good idea of how our system is behaving. So attached to metabolomics, we bring this to some of the critical quality attributes of biologics, and critical quality attributes are all of the attributes that make up that can affect the overall quality of your therapeutic, and so we want to know what kind of design space that we can use to be able to control that. Our lab, very similar to David Rose and Brendan, has a focus on glycosylation, especially on glycosylation of monoclonal antibodies, because we know that glycosylation can affect not only the secretion but the solubility, stability, and some of the therapeutic functions of proteins, including serum half-life and effector functions. One aspect of glycosylation and solubility that we're looking at is the ability to produce large quantities of protein in our system without precipitating out within the cell, which can be caused by unwanted glycosylation patterns. The other branch of work that we do in our lab is to try and control the when and how much of protein that we're producing. And this is, while the antibodies are primarily produced in mammalian cells such as CHO, we also work with insect cells. Insect cells are actually a very good system to produce large quantities of proteins that have very similar quality as human proteins, but we can actually produce them in very large quantities. They're being studied for the production of vaccine components, especially particles that resemble viruses that can be used in vaccines. And when you do this, you have multiple proteins that come together to form an end product. So in this case, a virus-like particle might be composed of multiple different types of proteins that come together and form, I guess, a little nanostructure. But we want to make sure that we can actually control the expression levels within the cell. And so what we've done is we started playing with promoter elements to try and tailor when expression is turned on and how much protein can actually get expressed. It's not a concept that hasn't been discussed in the literature, but it's a concept that hasn't been studied or hasn't been thoroughly investigated. And from an engineering point of view, if you think of all the work that's been done in characterizing flow in pipes to get all types of friction factors, well, there's a lot of basic work that's done there so that you can enable people to come along and design piping systems. We want to be able to do the exact same thing, but with protein expression. And so we're doing in-depth characterization of how different combinations of promoters can affect different protein expression levels. And so an example of this is at the bottom, where we can see that the ratio of two different proteins that we're expressing via the control of two different promoters can in fact affect how much of each protein is being expressed in cells. And so to conclude, our lab really focuses on this idea of complex biologics, whether it be something like a therapeutic antibody or a vaccine component like a virus-like particle or even viruses. And so we do a lot of work at trying to produce stuff. So we're a production lab trying to understand the production processes behind these biological systems. Thank you.